In this video, we will discuss about this extended analysis of mKRAS vaccination on patients. So what kind of patients undergoes this extended analysis? So the criteria that they have used is the patient who has already undergone some kind of treatment and later does not show any signs for the disease relapse, and these patients are agreed to undergo this extended mKRAS vaccinations at regular intervals. So those patients are being analyzed in these experiments, or the results that are presented in this figure. So there are like four candidates that fulfill this criteria, and those results are shown in this figure. So first they check the T cell activations through these IFN gamma signals, and they are able to see that there is some modest increase in the IFN gamma signals in all the four patients, and thereafter they go further deep into these T cells characterizations using the mass spectrometry that they have done in the previous experiments also. So there they have characterized these different T cells populations, that is CD4 central memory T cells, and they are able to see that compared to the control, these central memory T cells are increased in almost all the different mutant of mKRAS antigen. And similarly for the effector memory T cells also, they are able to see the increase in the signals for all the different mKRAS mutations compared to the control. And when you check the CD8 plus effector memory T cells, which is the main cells that kills the tumors, those signals are also significantly higher compared to the control, indicating that this mKRAS vaccination maintains the high level of T cells that prevent or that helps in relapse of the tumors after the treatment. And after that, what they did is, instead of directly checking the PBMCs of the patients, they cultured or in vitro cultured the PBMCs for extension period of time to this different mKRAS peptide, or this mKRAS vaccinations, and what they are able to see that there is a further elevated signals of this IFN gamma, or we can say that there is a further enhanced activation of the T cells has been observed in all the four patients. Though for this patient 3, that is J1994_2, the increase in signals is high for this G12R antigen and mKRAS antigens, but for other case it doesn't look like they are significantly higher. But in general, they have seen the increase in the antigens when they extended this PBMCs in vitro with these different mKRAS peptides. And then they checked within this CD4 plus T cells, there are different populations of CD4 plus T cells, such as this Th1 T cells, Th2 T cells, and Th17 T cells. There are other populations of the T cells also, but they characterize these three T cells through the cytokines that are released by these three T cells, and they are able to see that cytokines from this Th1 T cells, that is IFN gamma, IL-2s, are significantly higher in all the four patient case, whereas the cytokines for the Th2 T cells, that is IL-4, IL-5, are lower, but IL-10 signals are there to some extent, and they are higher in the patient second. But the signals for this Th17, that is IL-17A, they are very minimum in all the four cases. So this results indicates that not all the CD4 plus T cells populations elevated at the same level, but there is also preferential selections based on which the different T cells populations are expressed. Those selection criteria have not been discussed in this figure. We will see whether they have discussed how this preferential selection happens Whether they have discussed on that topic or not, we will see in our subsequent videos, where we will discuss the further main figures that are presented in this figure. So that's all for this video. I hope you understood the results for the extended analysis of mKRAS vaccinations and the criteria that they used to present the data, criteria that they used to filter the patients, and the data, how they interpret the data. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.